Hello, everybody. I wish everything is fine. In this lecture, we'll talk about how to choose the right sequences for multiple sequence alignment, for building multiple sequence alignment or MSA. Simply, in this lecture, we will see how to choose the right sequences. So before you build your alignment, of course, you have to select your sequences that you would like to be aligned. This sequence should be part, for example, from a protein family, and you would like to know the common ancestor for that family. So simply you should gather the members of that family, members of protein, I mean. Of course, the same thing apply to genes. And of course, you try to choose not too large to be entirely included in your multiple alignment. And picking the right sequences actually is very important, and it's a bit of art. I mean, it's like art. You have to do it very carefully and I mean, you have to choose it in a careful manner. So simply, you can just go for example, some websites like Uniprot. Here, you can retrieve sequences from uniprot.org, of course, and you can download it in a faster format. So simply go to the website, Uniprot website, and then choose like the arrow number one shows. Here, you can choose which one you would like to have. Like here, we choose rat, mouse, chicken, Arabidopsis, also human. And then in number two, we can just click download and then download the selected ones in faster format. And then you just press go like number four, the arrow number four shows. So this is the procedure of just retrieving our sequences from a website like Uniprot. The same thing can be applied for other databases like this American database, the NCBI. NCBI, you can also retrieve sequences from it. Simply press here, the R number one. We here choose what kind of sequences we want, like we just choose protein and then the name of the protein, here HSP70, and then arrow number two shows the what are the number of the members of the protein family that we would like to use from different organisms. Here we just choose to check whatever you want, and then number three here, the arrow number three shows that how we can download these sequences. Simply just press send to, which is here at number three, in our number three, and then we can just create files, of course, in a FASTA format. Choose here file, and then FASTA, the type of the file is a FASTA format, and then create. Then you can get your sequences downloaded, either from Uniprot or from NCBI. Both of them are a database, contain a lot of information and a lot of sequences that can be used. Uh, of course, we have to stick with some guidelines for selecting sequences. First of all, are we going to use protein or DNA? Which is better? Uh, I think always stick with protein whenever possible, because simply you can turn them back to into DNA, and then you can do the multiple before after doing the multiple sequence alignment. So usually it's much better to choose protein. If the sequences are not coding sequences, you must use DNA, of course. And you can do this very simply. Okay. Number two is how many sequences I can use. How many sequences I can use for multiple sequence alignment. You should start with from 10 to 15. Avoid aligning more than 50 in the beginning. So when you would like to start to do the multiple sequence alignment, it's much better to try to with a smaller set, like from 10 to 15. Of course, you can do less, of course, but to have informative uh, alignment, multiple sequence alignment, you should start with, for example, from 10 to 15. Okay. Question number three, I mean, too much differences. Should I use too much different sequences? No. Sequences that are less than 30% identical, two more than half the other sequences in the set often cause trouble. So don't use too much different sequences. Don't use sequences that are really too different or too much different. You just need something like not too much different and not too much identical as you see in my next slide. So what about identical? Should we use identical sequences? No, they will never help unless you have a very good reason to do so, or you are looking for something. So in this case, you can use identical sequences. But as I said, not too much, not like not too much identical and not too much is also different. So it's something in between. So it will be more uh, better for in this case. So avoid incorporating multiple sequence alignment sequences that are more than 90% identical and not less than 30%, also not less than 30% identical are also or similar and not more than 90% identical. Unless, and also I usually try to stress on this, unless you have a good reason to do this in your experiment. So this is very important. Also, you should not use partial sequences. Multiple sequence alignment program also prefer that roughly the same lengths or sequences that all the same lengths. So don't use partial sequences. So if you're going to use multiple sequence alignment in neural analysis, don't use partial sequence. So it's very difficult to compare with complete sequences and make many trouble. Okay. Also, to try to avoid sequences that have repeated domain. This also cause trouble in most program, and that's why if there is different domains, and these domains have in each sequence have a different number, so also this causing trouble. And in this case, it's better, as we said, always try to extract them off, extract your domain yourself using dotlet or lavine, and we will talk about them later. And making a multiple alignment of those segments, so you can extract these segments with your, of course, by yourself using this two program. If you would like to see this domain or work with sequences that have multiple domain and the number of domain are different across different sequences. So other thing also from the guidelines that if you cannot generate a proper alignment from sequences that you are related, I mean if you still cannot generate a proper alignment from the sequence that you are related, you could use a local multiple alignment method such as Gibbs sampler, pattern extraction motive, or PRET. So in this case, as you see, always try to have a group of sequences that are not too related or not too similar, not 90, but more than 90% similar or less than 30% identical. Okay, now we have PRET. PRET is one of the very good program that help you to extract motive using using this tool, and it's available in the API, which Impel API, European Molecular Biology Laboratory, in the European Bioinformatics Institute. So PRET is a very good tool for extracting motives. You can use, I mean, you can easily use it and just paste your sequence here if you would like to extract some of the motives from your sequences. For carrying out a phylogenetic tree, phylogenetic analysis on a set of coding DNA sequences, do the following. First is translate your DNA into proteins. As I said to you, it's much, much better. Perform multiple sequence alignment on the protein. Thread the DNA back onto the protein, multiple sequence alignment. And as you see, you have two very important programs that you can use. Simply, you can use Paltonel. It's very important and easy program to use. Also, you have Protogen. You can do the same thing for you. So both programs can help you in this application. And then you can, of course, thread the DNA back onto the protein multiple sequence alignment framework using Protogen. This is I just give you the paper in arrow number one, just show you the paper published for this program, Protege. Okay. First of all, if you would like to choose the right number of sequences, as we said in the beginning, it's from 10 to 15. This is good for a start, suitable for most cases. For multiple sequence alignment, this is the right number. After you get something, of course, interesting happening, you can increase the size, and it's hard it's hard to see any reason to have like more than 50. Unless of course you have interested to build an extensive phylogenetic tree. So it's much better if you would like to make a multiple sequence alignment and build a phylogenetic tree later on, to start with a small set of sequences from 10 to 15. Then you can increase if you need that, if it's needed. If not, that will be, I think, a suitable number for making this. Also, we have, if you have a big number of sequences and you try to align them, there is a lot of trouble that is related, will be related with the bigger number of sequences in your multiple sequence alignment. Number one is computing. Computing will be difficult with the bigger number. Also, building a big alignment is difficult, take time. Also, displaying them in your screen will be also a problem, will be difficult. Also, using big alignment, also difficult. So difficult, and making accuracy with this big alignment is difficult. So you will have problem with computing, building, displaying, using, and accuracy. This five problem could be associated with uh, having much sequences in to align in your multiple sequence alignment. Also, you choose the should you choose sequences that are very similar or very different? This is a very good question. Some people ask me, what kind of sequences should I choose? Should I choose the very similar or very different? Of course, as you said, make the right compromise between similarity and new information. So first, you have to have your sequences related, but not too similar. And as you said, less than 30% identical is not allowed, and more than 90% identical is not preferred unless you have a good reason to do so. An alignment that contain very similar sequences brings later information, as we said. You can use it to extrapolate annotation, but you cannot do phylogeny, structure prediction, functional prediction by these kind of sequences or any of the other useful application that we mentioned before. So, as I said to you, you can always choose sequences that are not very identical or similar and not very different, as we said before. General rule is simply, the general rule is that you want them to be as distantly related as possible, also without requiring too many gaps in order to be properly aligned. So this is the general rule for choosing your sequence. And you have always to keep this in mind. Two things multiple sequence alignment program really don't like are, number one is sequence that are very different from every other sequence in the group. This is make a problem. This criteria is, or this number one is this make a big problem in the alignment, and you will feel that the multiple sequence alignment is a lot messy. Is a very bit messy, a bit messy with this. If you have a sequence that very different from the other sequences in the alignment. Number two is sequences that need long insertion uh, or deletion to be properly aligned. That you will see some of the sequences, you just need to put yourself some insertion, some deletion. You have to make a lot of sequence change in the sequence to make it properly aligned with other sequences. In this case, really, also this kind of sequences make problem with any program. not just specific program. Let's talk now about gathering sequences with online class servers. Of course, as we said, you have two options to collect sequences that are characterized sequences or uncharacterized sequence. First of all, characterized sequence always is good for annotation if you are working with good annotation and experimental information. And this all can bring this biological information when you make a multiple sequence alignment with this kind of sequence, with the characterized sequence, it brings the biological information with them to allow, of course, feature propagation. So that's why we like this characterized sequence to be used when you're making a multiple sequence alignment. Okay, sometimes you need to have some uncharacterized sequence, and we can conclude them only to distinguish between the conserved position and position that cannot be mutated. Also, it's less, and also you can get some more information would be helpful to discover many things in this uncharacterized sequence because, as you say, it may help getting more contrast on your sequence of interest. So that's why we use both sequences. Should have characterized sequence and also uncharacterized sequence, but just try to understand why we use this and why we use that. So sequences that are so similar to the query are properly homologs. Of course, here we just say this usually that sequences that are so similar to the query maybe homolog having the same ancestor. So we commonly refer as hits or matches. So this is the name you will find it in the database, hits or matches. So what we're looking for, we're looking for homologs of your sequence. If you're doing search using class, for example, and these hits or matches are the homologs for your sequence that could be used, as we said, in the multiple sequence alignment. Of course, now we will just tell you about some programs that can be used very easily and the difference between them. We'll talk about them later in the coming lectures. First, we'll talk about cluster omega. It's one of the commonly used one, and usually everybody use it. And it's this program, of course, as we said, it have the يعني mean that use what you call hidden Markov model or HMM profile techniques to make this generate the alignment. And this is the name of the algorithm, hidden Markov model that this program use. It's called cluster omega. You can use it. It is of course one of the European program. You can find it in the API very easily. And by the way, the Uniprot using that program, cluster omega, and it's preferred for aligning protein. Okay, the other program is called tcaffe, and it's preferred for small alignment, and it can be used for protein, RNA, or DNA. And also, it's a very important program, um, and uh, and that the name tcaffe and is preferred, as we said, good for small alignment. Also, we have another one which called muscle. It's good for medium size alignment and one of the fastest alignment method around. And it's good for large size alignment, but as I said, medium size. And when we say medium size, it means that more than 500. And small means less than 500. And when we say bigger than this, we'll say like there is a program called MAFT, and MAFT can be used for a bigger alignment. Of course, MAFT is a large size alignment. It can be used more than 5,000 alignment. I mean, it can be used for 5,000 sequence. So we have now three different categories. For small alignment, uh, uh, TCAFE is good. For medium alignment, which is more than 500 muscle is good. For large alignment, more than 5,000 MAFT is good. So you have the three different categories. So Cobalt is a newcomer actually, and not just, I mean, new doesn't mean newcomer comparing to the others. And it's good for multiple sequence alignment of routines, and of course using conserved domain and local sequence similarity information. This is available in NIH or NCBI, and why we mention this? Because actually most of the program we just mentioned before, like cluster omega, muscle, MAFT, and TCAFE, all of them are European programs. Actually, Cobalt is the only one that is belong to the NCBI and the National Center of Biotechnology Information. So it's a newcomer comparing to the others. Okay, so multiple sequence alignment, it can build, of course, there is some programs that can be used to build a multiple sequence alignment like this one. We'll talk about in the next module. GalView. GalView is a very important and very good and very useful multiple sequence alignment. It has a website, and it also you can download in your computer, and you can use any of the algorithm we just mentioned. It has TCAFE, it has PropCons, it has muscle, it has MAFT, it has props, MSA props, it has props, cluster, and cluster omega in one package, and also it has many other useful feature. I will talk about this program in our next video. Multiple sequence alignment also, as we see, could also available also in other program like Mega, and this program use two important multiple sequence alignment, cluster W and muscle. Both these two program available in the Mega. So this is two tools for multiple sequence alignment available in Mega program. Now you can try in your own time. Just open the link, and then this link will lead you to a multiple sequence, multiple I mean questions about multiple sequence alignment and all that. You can use in your try in your own time, and this will help you to understand this topic and also answer some question and practice by answering some of those questions. Thank you very much, and let's go to the next lecture.